Hi, everyone. Thank you for keeping your subscription to this channel. I'm here again to bring to you interesting and fascinating discovery in science. This discovery was published in an article recently, and I personally found it interesting, and I would like to share it with my audience in this on this particular channel. So it is a discovery that challenged the central dogma of molecular biology. For those who are not familiar with this term, the central dogma of molecular biology describes the flow of information from DNA to RNA and to final molecule, which is called proteins. So in molecular biology, genetic information flows is believed to flow from DNA until we have the final product, which is a protein. Several processes are involved in this flow of information, some of which are well studied. One of them, which is like the first one, is transcription. It is the change or the flow of information from DNA to RNA. And another one is translation, which is the conversion of information on the RNA into a functional protein, which becomes like the functional molecule. And even in past years, it has been described how information on RNA can be converted back to DNA by a process called reverse transcription. So, but the gap in this field of knowledge is that no one has ever reported the synthesis or the reverse flow of information from protein to nucleic acid molecule, either to DNA or even to RNA. In the specific course that I took in my current studies, the lecturer was inquiring what we think about a process that converts proteins back to DNA. One, as a person, one would think that such process would be somehow expensive in the biological system to change protein to DNA. Such process itself would be energy demanding. At the same time, it will require a great precision to be able to achieve that kind of process within the cell. It is quite interesting that a recent study suggests that this process might be feasible in the biological system, and that is what is very interesting and what I have found very fascinating. So to describe briefly, two things are very important for DNA synthesis, which includes the DNA primer and a DNA template. These two things are very important for DNA synthesis to initiate and also to continue, for DNA polymerization to continue in the appropriate sequence the cell wants it. To give a brief history that led to this recent discovery, in 1983, a group from University of Arizona in the US observed that the DNA synthesis or DNA replication in this particular phase is primed by a protein. So instead of the normal DNA or nucleic acid priming of DNA synthesis, they observed that the replication process in this particular bacteriophage is primed by a protein. This finding is the starting point for the recent discovery. So in 2011, researchers from the University of Calgary in Canada reported some proteins which are related to reverse transcriptase that have this similar property of protein priming at the same time and untemplated DNA synthesis. It implies that these proteins, which are related to reverse transcriptase, they can synthesize DNA with a protein serving as a primer and without a DNA template. So that also is very significant in the history behind the recent discovery. Although in 2011, the mechanism behind this observation was not that elaborate, but later in 2022, a paper published by a group from Poland described the mechanism behind this protein primed and untemplated DNA synthesis observed for these families of protein, which I've said are related to reverse transcriptase. And last month, April 16 particularly, a paper was published by a particular group from the University of Stafford describing a difference-associated reverse transcriptase named DRT3A, capable of carrying out a protein primed and protein templated DNA synthesis to produce. And 3 days after that, another paper, but not yet published, another manuscript was released on Biorex describing another type of that same families of proteins, DRT7, that is also capable of producing palindromic DNA containing A and T's. These findings are very important in molecular biology field, and they are new discoveries that has really come to challenge the central dogma of molecular biology. So the summary of it is that we now know that there are proteins that are capable of synthesizing DNA from a protein template. This discovery for me is interesting. This discovery for me is fascinating, and I believe maybe someday this discovery is going to win a Nobel Prize. So thank you for listening to my history, but I hope that you find this very interesting, because this is a finding that will initiate a new, potentially a new field of research, and at the same time several future applications even. So thank you for watching this particular video. Thank you for staying tuned, and I hope to bring to you more interesting and fascinating discoveries in science. Thank you. Bye.